Hello everyone, welcome to today's presentation. I'm excited to have you here. Today we'll be exploring excretory products and their elimination. This is a fascinating topic that covers how our bodies manage waste and maintain balance. Over the next few slides, we'll look at the different types of excretory products, how they are eliminated, and why this process is so vital for our health. Let's get started. Let's now look at disorders of the excretory system and their diagnostic approaches. One important aspect is urine testing, which plays a crucial role in diagnosing various kidney and urinary tract conditions. Moving on, we see the significance of the heart, specifically the atrial natriuretic factor, or ANF, which helps regulate blood pressure and fluid balance by influencing the kidneys. In our summary, we focus on the mechanism of concentration of the filtrate within the nephron, which is essential for urine formation. The nephron is the functional unit of the kidney, and it works through complex processes to produce urine. We also explore the excretory organs across different organisms, starting with simpler structures like protonephridia or flame cells, which are found in some invertebrates. These are involved in early forms of excretion. Next, we delve into modes of nitrogenous waste excretion: aminotolism, ureotolism, and urocotolism. These are strategies organisms use depending on their environment and evolutionary adaptations. We then examine the role of the juxtaglomerular apparatus, or JGA, and its importance in the renin-angiotensin system, which helps regulate blood pressure and kidney function. The countercurrent mechanism is another key concept that enables the kidney to concentrate urine efficiently, maintaining osmolarity and osmoregulation. Osmoregulation is vital for maintaining water and salt balance, and the kidney plays a central role here. Inside the kidney, processes like hemodialysis are used to assist individuals with renal failure. Other excretory structures include antennal glands in arthropods and Malpighian tubules in insects, showing the diversity of excretory adaptations across the animal kingdom. Micturition, or urination, is the process of expelling urine, which is controlled by complex mechanisms involving the bladder and nervous system. We also discuss the regulation of kidney function through hormones like ADH from the hypothalamus, which controls water reabsorption, and other organs that assist in excretion. The structure of the nephron, including the function of its various parts, such as the tubules and the vasa recta, is fundamental for understanding how urine is formed and concentrated. Finally, topics like kidney transplants and the importance of understanding these mechanisms are highlighted, emphasizing their relevance in medical science and patient care. Thank you for your attention. Today, we're going to explore the concept of excretion, which is essentially the process by which animals eliminate metabolic waste products from their bodies. This process is crucial because it helps regulate the composition of body fluids and tissues, maintaining a healthy internal environment. Now, the waste products that need to be excreted include substances like ammonia, uric acid, urea, and carbon dioxide. Each of these has a specific role and importance, depending on the animal's physiology. In addition to these, ions such as sodium, Na+, calcium, Ca+, chloride, Cl-, as well as phosphates and sulfates, are also eliminated through excretion. These ions are vital for various bodily functions, so managing their levels is essential for overall homeostasis. As we move through this chapter, keep in mind that excretion isn't just about getting rid of waste. It's about maintaining the delicate balance of chemicals and fluids in the body, which is fundamental to the health of all living organisms. Let's take a closer look at aminotolism, which is the excretion of nitrogenous waste primarily as ammonia. Ammonia is highly toxic, so animals that excrete it need to do so in large volumes of water to dilute it effectively. This method is common in aquatic animals like fish. Moving on, urea is another major nitrogenous waste product. It's less toxic than ammonia and is quite water-soluble, allowing animals like mammals and amphibians to excrete it with a moderate amount of water. This process is called ureotolism. Then there's uric acid, which is the least toxic among these waste products. Because it's relatively insoluble, animals can excrete uric acid with minimal water loss. This is especially advantageous for terrestrial animals like reptiles and birds, helping them conserve water. This process is known as uricotylism. To summarize, the major forms of nitrogenous waste excreted by animals include ammonia, urea, and uric acid. Each has its own biological advantages depending on the animal's environment and water availability. This diversity of excretion strategies highlights how vital excretion is for maintaining internal balance and overall health. Let's take a closer look at the different modes of excretion based on the nature of nitrogenous waste. As organisms have evolved, they've developed various ways to eliminate these wastes efficiently. The key point here is that the type of nitrogenous waste an organism excretes depends largely on its environment and its metabolic needs. For instance, some animals excrete urea, which is less toxic and requires moderate amounts of water to eliminate. This is common in terrestrial animals like mammals and amphibians. Then we have animals that excrete uric acid, which is even less soluble in water, allowing them to conserve water. Think of birds and reptiles. Finally, there are animals that excrete ammonia directly, which is highly toxic, but requires a large volume of water to safely dilute it. This is typical for aquatic animals like fish. Understanding these modes helps us appreciate how different animals adapt their excretory systems to their habitats and lifestyles, connecting directly to the overall theme of how organisms maintain internal balance and homeostasis in diverse environments. Ammonia, as it is readily soluble, is generally excreted by diffusion across body surfaces or through gill surfaces, especially in fish, as ammonium ions. Many bony fishes, aquatic amphibians, and aquatic insects rely on this method. The process of excreting ammonia is called ammonotylism. Animals that excrete most of their nitrogen in the form of ammonia are known as ammoniotic. This mode of excretion is efficient in aquatic environments where ammonia can easily diffuse away due to the surrounding water. It's a key adaptation for aquatic animals to manage their nitrogen waste effectively. Now, let's take a closer look at some animals that utilize uricotylism. This includes mammals, many terrestrial amphibians, and marine fishes. These animals share a common strategy for excreting nitrogenous waste. When these animals metabolize proteins and other nitrogen-containing compounds, they produce ammonia, which is quite toxic. To handle this, their bodies convert ammonia into a less harmful substance called urea in the liver. This urea then enters the bloodstream, where it is transported to the kidneys. The kidneys filter the blood, and during this process, urea is separated out from the blood plasma. The filtered urea is then excreted out of the body in urine. This entire process of converting ammonia into urea and excreting it is called uricotylism. Animals that excrete urea are known as ureotic animals. This method is especially advantageous for terrestrial animals because urea is less toxic than ammonia and can be safely stored temporarily in the body before excretion, making it an efficient way to manage nitrogen waste in different environments. So, in the context of our overall discussion on excretion strategies, uricotylism is a key adaptation that balances toxicity, water conservation, and the needs of terrestrial and marine life. Now, let's look at some examples of animals that excrete uric acid, including reptiles, birds, land snails, and insects. These animals have adapted to conserve water by excreting uric acid in the form of a pellet or paste, which minimizes water loss. This method is particularly advantageous for animals living in dry environments. The process of secreting uric acid is called uricotylism, and animals that excrete nitrogenous wastes this way are known as uricotic animals. This mode of excretion is a key adaptation for water conservation and is a crucial part of how these animals survive in their respective habitats. Most invertebrates have a relatively simple excretory system, featuring a tubular structure known as protonephridia or metanephridia. These are considered primitive kidneys and are quite effective for their simpler bodies. In contrast, vertebrates have evolved much more complex tubular organs called kidneys, which perform more advanced and efficient waste management. This difference highlights how excretory structures have become more sophisticated as animals increased in complexity. Understanding these variations helps us see the connection between an animal's body plan and its excretory system, emphasizing how essential waste removal is for maintaining internal balance across all animal species. Let's take a closer look at Protnephridia, also known as flame cells, which are the main excretory structures in platyhelminthes, like flatworms such as planaria. These structures are also found in some rotifers and certain annelids. Protnephridia are specialized for managing the organism's internal environment, primarily focusing on ionic balance and fluid volume regulation, which is a key aspect of osmoregulation. Essentially, they help the organism maintain the proper balance of salts and water, which is vital for survival. By doing so, they ensure the organism's internal conditions stay stable despite changes in the external environment. This function is crucial in aquatic environments where water and salt levels can fluctuate considerably. So, in the context of the overall theme of excretion and osmoregulation, Protnephridia play a fundamental role in helping these animals maintain homeostasis. Nephridia are fascinating structures because they serve as the tubular excretory organs in earthworms and other annelids. These tiny but vital organs play a crucial role in helping the organism get rid of nitrogenous wastes, which are byproducts of metabolism that need to be expelled to maintain health. But their role isn't just about waste removal. Nephridia also help maintain the fluid and ionic balance within the body, which is essential for the worm's overall homeostasis. Think of them as the body's internal cleaning and balancing system, ensuring that everything stays in harmony as the earthworm moves through its environment. This function is a great example of how specialized structures support the survival of different organisms, fitting into the bigger picture of how animals adapt their excretory systems to their environment. Now, let's take a closer look at the Malpighian tubules, which are the main excretory structures in most insects, including cockroaches. These tubules play a crucial role in helping insects manage their waste, particularly nitrogenous wastes, which are byproducts of protein metabolism. They also assist in osmoregulation, maintaining the proper balance of water and salts within the body. Essentially, these tubules are vital for the insect's survival, ensuring waste removal and internal balance. Understanding these structures helps us see how insects have adapted to efficiently regulate their internal environment, which is key to their success in various habitats. Now, let's take a look at the antennal glands, also known as green glands, which are fascinating structures in crustaceans like prawns. These glands are specialized organs that play a crucial role in excretion. Essentially, they help remove waste products from the animal's body, maintaining internal balance and health. You can think of them as the crustacean's version of kidneys. They are located near the antennae, which is why they are called antennal glands. Their position is strategic, allowing them to efficiently filter out waste from the hemolymph, or the crustacean's blood equivalent. This excretory function is vital, especially because crustaceans often live in environments where waste buildup can be problematic. So, in summary, these glands are key to the crustacean's ability to regulate its internal environment and stay healthy in its aquatic habitat. Now, let's take a closer look at some of the key excretory organs found in various organisms. Starting with the Malpighian tubules, these are specialized structures in insects that function to remove nitrogenous wastes and help in osmoregulation. They play a crucial role in maintaining the organism's internal balance. Moving on, nephridia are excretory structures present in many invertebrates like earthworms, helping to eliminate waste while conserving essential substances. The kidneys, of course, are the most well-known excretory organs in vertebrates, responsible for filtering blood, removing wastes, and regulating water and electrolyte balance. Then there are antennal glands, which are found in crustaceans like crabs and lobsters. These glands help in osmoregulation and waste removal through their antennae. Proteinephridia are simpler structures typically found in flatworms, consisting of a network of tubules ending in flame cells that help eliminate excess water and waste. Overall, these different organs highlight the diversity of excretory systems across various species, each adapted to their specific needs, but all sharing the fundamental goal of waste removal and maintaining internal homeostasis. Now, let's take a closer look at the excretory system. It consists of a few key components that work together to remove waste from our bodies. First, we have a pair of kidneys, which are vital because they filter blood and produce urine. Then, there are the ureters, one for each kidney, that carry the urine down to the next part of the system. This next part is the urinary bladder, a storage tank that holds urine until we're ready to eliminate it. Finally, there's the urethra, which is the passage through which urine exits the body. Understanding how these parts connect and work together helps us appreciate how the excretory system maintains our body's balance and keeps us healthy. It's a great example of how our body has specialized structures that perform essential functions seamlessly. Now, let's take a closer look at the internal structure of the kidneys. At the core, we have the nephrons, which are the essential functional units responsible for filtering blood and producing urine. Each nephron connects to a calyx, which is part of the collecting system that funnels urine toward the renal pelvis. The cortex is the outer region of the kidney, where many of these nephrons are located, while the medulla is the inner part that contains the renal pyramids. The hilum is the entry and exit point on the kidney where blood vessels, nerves, and the ureter connect. Moving to the external structure, the kidneys are the main organs of the urinary system. The ureters are tubes that carry urine from the kidneys down to the bladder, which stores urine until it's ready to be expelled. The urethra then provides the passage for urine to leave the body. Understanding these structures helps us appreciate how the human urinary system functions as a whole, filtering blood, regulating fluids, and maintaining overall homeostasis. Let's take a closer look at the structure of the kidney. The outer layer of the kidney is protected and supported by three main layers of tissue. These are the renal fascia, which is a connective tissue layer that helps hold the kidney in place, the perineal fat capsule, a cushion of fat that provides insulation and protection, and the fibrous capsule, a tough layer that directly covers the kidney's surface. Moving inward, on the inner concave side of the kidney, there's a notable notch called the hilum. This is a crucial entry and exit point where the ureter, blood vessels, and nerves pass into and out of the kidney, facilitating its vital functions. Just beneath the hilum, you'll find the renal pelvis, a broad, funnel-shaped space that collects urine from the kidney. From here, urine is funneled into the ureter. Inside the renal pelvis, there are projections called calyces, which are like small chambers that collect urine from the renal papillae before it drains into the pelvis. Now, let's talk about the physical characteristics of the kidneys. They are reddish-brown and bean-shaped structures, positioned between the last thoracic vertebra and the third lumbar vertebra, close to the dorsal inner wall of the abdominal cavity. In terms of size, each kidney in an adult typically measures about 10 to 12 centimeters in length, 5 to 7 centimeters in width, and 2 to 3 centimeters in thickness. Their average weight is roughly between 120 and 170 grams. Understanding these structural features helps us appreciate how the kidneys efficiently perform their vital role in filtering blood and maintaining overall fluid and electrolyte balance within the body. Now, let's look inside the kidney. The medulla is organized into a few conical structures called medullary pyramids. These pyramids project into the calyces, which are essentially the chambers that collect urine. Between these pyramids, you'll find the cortex extending outward as what we call the columns of Burton, or renal columns. These columns of Burton serve as a kind of bridge, supporting the structure of the kidney and allowing blood vessels and nerves to navigate between the cortex and medulla. If we think about the overall architecture, the kidney is divided into two main zones: an inner medulla, where these pyramids are located, and an outer cortex, which surrounds them. Understanding this internal organization helps us appreciate how the kidney efficiently filters blood and concentrates urine, all within a highly specialized and compartmentalized structure. The functional unit of the kidney is called the nephron. Each kidney contains about 1 million nephrons, which highlights just how complex and efficient our kidneys are. Now, every nephron is made up of two main parts, the glomerulus and the renal tubules. The glomerulus acts as the filtering unit, where blood is filtered to begin the process of urine formation. The renal tubules then carry this filtered fluid, reabsorbing what the body needs and secreting waste products. Together, these parts work seamlessly to maintain our body's fluid balance, electrolyte levels, and waste removal, all essential for overall health. Understanding the nephron really helps us see how vital the kidney's role is in maintaining homeostasis. Let's take a closer look at the structure of the nephron, starting with the glomerulus. The glomerulus is essentially a tuft of tiny capillaries, and it's formed by an afferent arteriole, which brings blood into this network. From there, the blood is filtered through these capillaries, allowing the kidneys to perform their vital function of filtering waste and excess substances from the blood. Once the blood has been filtered, it exits the glomerulus via the efferent arteriole. This part of the process helps regulate blood pressure within the glomerulus and directs the filtered fluid into the next segment of the nephron. Moving on, the renal tubules begin with the Bowman's capsule. This capsule surrounds the glomerulus and captures the filtrate, which will then undergo further processing. The renal tubules are divided into several parts. The proximal convoluted tubule, where reabsorption of essential substances begins, Henle's loop, which plays a crucial role in concentrating urine, and the distal convoluted tubule, where fine-tuning of the filtrate occurs. It's important to remember that the Bowman's capsule and the glomerulus together form what we call the renal corpuscle. This is the initial site of blood filtration in the nephron, setting the stage for the complex process of urine formation. Understanding this structural organization helps us see how each part of the nephron contributes to maintaining the body's fluid and electrolyte balance, which is central to kidney function and overall homeostasis. Now, let's take a closer look at the juxtamedullary nephrons. These nephrons are quite distinct in their structure and function. The key point here is that the Malpighian tubules, along with the proximal convoluted tubule and distal convoluted tubule, are primarily situated in the cortical region of the kidney. However, what really sets juxtamedullary nephrons apart is that their loops of Henle extend much deeper into the medulla. This extension allows them to play a crucial role in concentrating urine effectively. In contrast, most nephrons, about 85%, are called cortical nephrons. Their glomeruli are located in the outer cortex, and their loops of Henle are relatively short, only extending a little into the medulla. They are also smaller in size compared to juxtamedullary nephrons. So, to summarize, the juxtamedullary nephrons, although fewer in number, have longer loops of Henle that go deep into the medulla, making them essential for water conservation. The cortical nephrons, on the other hand, are more numerous and have shorter loops, mainly involved in filtration and initial processing. Both types are vital, but their structural differences underpin their specific functions in the kidney's overall ability to regulate water and solutes. Now, let's talk about the vasa recta, which are specialized blood vessels associated with the nephron. After the efferent arteriole exits the glomerulus, it forms a network of capillaries known as the peritubular capillaries. These capillaries surround the renal tubules and are crucial for the kidney's function. A small vessel of the peritubular capillaries runs parallel to Henle's loop, and this structure is called the vasa recta. The vasa recta forms a U-shaped loop, which is essential for the kidney's ability to concentrate urine through a process called countercurrent exchange. This countercurrent mechanism allows for efficient reabsorption of water and solutes, helping the kidney conserve water when needed. Interestingly, in cortical nephrons, which are the majority of nephrons, the vasa recta is either very reduced or absent altogether. Understanding the vasa recta is important because it highlights how the kidney maintains the body's fluid and electrolyte balance, which is central to its overall function and our health. All right, let's talk about diffusion and facilitated diffusion. These are forms of passive transport, meaning they don't require energy. They allow substances from the filtrate to move back into the body, a process known as absorption or reabsorption. It's like the body reclaiming valuable nutrients or molecules it needs. Then we have active transport, which is a bit different because it consumes energy, specifically ATP. This process moves substances into the filtrate when necessary, such as during secretion. Secretion helps the body get rid of waste or regulate substances more precisely. So overall, these processes, diffusion, facilitated diffusion, active transport, and secretion, are essential for maintaining the body's balance. They work together to ensure that the right substances are absorbed, reabsorbed, or secreted at the right times, keeping our internal environment stable. Let's take a closer look at urine formation. This process is fundamental to how our bodies maintain balance and remove waste. It all begins with the kidneys, which filter the blood to create urine. The kidneys perform this complex task through several stages, starting with filtration in the glomeruli. Here, blood pressure pushes water and small molecules out of the blood and into the renal tubules, forming what's called filtrate. Next, in the tubules, the body selectively reabsorbs essential substances like glucose, certain ions, and water, ensuring that valuable nutrients are conserved. At the same time, waste products such as urea and excess ions are left behind or secreted into the tubules. This fine-tuned process helps regulate blood volume, blood pressure, and electrolyte balance. Finally, the remaining fluid, now called urine, moves into the collecting ducts, where further concentration can occur. The urine then travels through the ureters to the bladder for storage until it is expelled. Understanding urine formation highlights how the kidneys serve as vital filters, balancing our body's internal environment. It's a remarkable system that connects directly to our overall health and homeostasis. Now, let's talk about the first step in urine formation, which is called glomerular filtration. This process involves the blood being filtered by the glomerulus, a tiny but vital part of the kidney. On average, about 1,100 to 1,200 milliliters of blood are filtered every minute. To put that into perspective, that's roughly 1/5 of the blood pumped out by each ventricle of the heart each minute. The driving force behind this filtration is the blood pressure within the glomerular capillaries. This pressure pushes the blood through 3 key layers. First, the endothelium of the glomerular blood vessels. Second, the specialized epithelium of Bowman's capsule, which includes cells called podocytes. And third, a basement membrane that lies between these 2 layers. Understanding this layered structure is crucial because it explains how filtration selectively occurs, allowing water and small molecules to pass through while larger components, like blood cells and proteins, stay in the bloodstream. This step is fundamental because it sets the stage for the entire process of urine formation, helping the kidneys filter waste products and maintain overall fluid balance in the body. Let's take a closer look at how the podocytes are arranged. They form this intricate network that creates tiny filtration slits or slit pores. These structures are essential because they allow blood to be filtered very finely through the membranes. As a result, almost all the components of plasma, except for proteins, pass through these slits and enter the lumen of Bowman's capsule. This selective filtering process is what we call ultrafiltration. It's a crucial step in the overall process of kidney function, helping to filter blood efficiently while retaining important proteins in the bloodstream. This slide highlights the importance of the specialized structure of podocytes in enabling this precise filtration. The juxtaglomerular apparatus, or JGA, is composed of the distal convoluted tubule and the afferent arteriole. This structure plays a crucial role in regulating kidney function. One of the key concepts here is the glomerular filtration rate, or GFR, which is the amount of filtrate the kidneys produce every minute. Typically, this is about 125 milliliters per minute, which adds up to roughly 180 liters per day. That's an incredible volume being filtered constantly to maintain our body's balance. Now, controlling this rate is vital because it ensures the kidneys are working efficiently without overworking or underperforming. The JGA is responsible for this regulation. When GFR drops, the JGA cells sense this decrease and respond by releasing renin. This hormone then stimulates changes that increase blood flow through the glomerulus and bring the GFR back up to normal levels. So, in essence, the JGA acts as a critical control point, maintaining the delicate balance of filtration, which is fundamental to overall kidney function and, ultimately, to our health. This mechanism highlights how tightly the kidneys regulate their activity to support homeostasis. Now, let's take a closer look at the volume of filtrate that forms each day. Typically, about 180 liters of filtrate are produced daily. However, what's really interesting is that only around 1.5 liters of urine are actually released from the body. This means that roughly 99% of the filtrate must be reabsorbed by the renal tubules. This crucial process is called reabsorption, and it's essential for maintaining the body's fluid and electrolyte balance. Essentially, the kidneys are extremely efficient. They filter a large volume of fluid, but most of it is returned to the bloodstream. Understanding reabsorption helps us appreciate how the kidneys regulate what stays in the body versus what gets excreted. It's a key step in ensuring that our internal environment remains stable, which ties directly into the overall function of the renal system. Let's talk about how the kidneys handle nitrogenous wastes and water. These substances are processed through different mechanisms. Some are passive, others active. Passive reabsorption involves substances like glucose, amino acids, and sodium ions. Plus, these are reabsorbed mainly through diffusion or facilitated diffusion, depending on the substance. Active reabsorption, on the other hand, requires energy. It's carried out by tubular epithelial cells located in various segments of the nephron. These cells use active transport mechanisms to move substances against their concentration gradients. The overall process is called reabsorption, and it's crucial because it allows the kidneys to reclaim valuable molecules and water from the filtrate back into the blood. This ensures our body maintains proper balance and prevents the loss of essential nutrients. Now, let's look at the next step in kidney function: secretion. The tubular cells play a crucial role here by secreting substances like hydrogen ions, potassium, and ammonia into the filtrate. This process of tubular secretion is vital because it helps us maintain the proper ionic balance and acid-base balance in our body fluids. Essentially, it's how the kidneys fine-tune the composition of our blood and urine, ensuring everything stays in equilibrium. This step highlights the kidney's remarkable ability to regulate our internal environment, supporting overall health and homeostasis. Now, let's take a closer look at one of the two types of nephrons, specifically the juxtamedullary nephrons. These nephrons are particularly interesting because of their unique location and function within the kidney. They are situated closer to the medulla, which is the inner part of the kidney, and their long loops of Henle extend deep into the medulla itself. This anatomical feature is crucial because it allows them to play a significant role in concentrating urine. By having these extended loops, juxtamedullary nephrons help establish the osmotic gradient necessary for water reabsorption, which conserves body water and maintains overall fluid balance. This specialization is essential for animals and humans that need to adapt to varying levels of hydration and electrolyte balance. So, in the context of our overall discussion about kidney function, juxtamedullary nephrons are key players in the body's ability to produce concentrated urine, making them vital for maintaining homeostasis under different physiological conditions. Now, let's take a closer look at the components involved in the nephron's structure and function. When we talk about the tubules, we're referring to the PCT, Henle's loop, DCT, and collecting duct. These are the main pathways through which filtrate passes, and each segment plays a critical role in reabsorption and secretion. Moving on to the renal corpuscle, it includes the glomerulus and Bowman's capsule. This is where the initial filtration of blood occurs, forming the filtrate that will eventually become urine. It's a finely tuned system that balances blood flow and filtration rate. The renal corpuscle is connected to the afferent arteriole, which brings blood into the glomerulus. Interestingly, the DCT, or distal convoluted tubule, is also involved in regulating blood pressure and electrolyte balance. Finally, the juxtaglomerular apparatus, or JGA, is a specialized structure located near the glomerulus. It plays a key role in regulating blood pressure and filtration rate by releasing renin. This feedback mechanism ensures that the kidney maintains homeostasis effectively. Together, these components form an integrated system that manages blood filtration, volume, and composition, core themes in understanding kidney function and overall fluid balance. All right, let's take a closer look at the functions of these different parts of the nephron tubules. Starting with the proximal convoluted tubules, or PCT, this is where a lot of the reabsorption happens. The PCT reabsorbs essential nutrients, water, and ions back into the bloodstream, which is crucial for maintaining the body's balance. Moving on to the loop of Henle, this structure creates a concentration gradient in the kidney, which allows the kidney to produce urine that is more concentrated than blood. This is key for conserving water. Then we have the distal convoluted tubule, or DCT, which fine-tunes the reabsorption and secretion processes. It helps regulate sodium, potassium, and pH levels, adapting to the body's needs. Finally, the collecting duct, or CD, acts as the final site for water reabsorption, influenced by hormones like ADH. It also plays a role in the excretion of waste products. Altogether, these segments work together to filter blood, reabsorb what the body needs, and excrete waste efficiently, ensuring overall fluid and electrolyte balance. Let's take a closer look at the lining of the proximal convoluted tubule. It's lined by a simple cubital epithelium with a brush border, which is essentially tiny microvilli that greatly increase the surface area. This expanded surface area is crucial because it enhances the tubule's ability to reabsorb nutrients and other vital substances from the filtrate. Now, most of the important nutrients, think glucose, amino acids, and a significant portion, around 70 to 80%, of electrolytes and water are reabsorbed here. This process is essential for conserving the body's resources and maintaining homeostasis. Additionally, the proximal tubule plays a key role in maintaining the body's pH and ionic balance. It does this through selective secretion of hydrogen ions, H+, ammonia, NH3, and potassium ions, K+, into the filtrate. At the same time, it absorbs bicarbonate ions, HCO, from the filtrate, which helps buffer the blood and keep the pH within a healthy range. And speaking of the proximal tubule specifically, this is the first segment in the nephron where a lot of reabsorption and secretion happen, making it fundamental to the kidney's function of filtering and balancing bodily fluids. Now let's look at the two key regions of Henle's loop, the descending and ascending limbs. The descending limb is special because it's permeable to water, but almost completely impermeable to electrolytes. As the filtrate moves down this limb, water exits into the surrounding medullary interstitium, which helps concentrate the filtrate. On the other hand, the ascending limb is quite the opposite. It's impermeable to water, so no water leaves here, but it actively or passively transports electrolytes out of the tubule. This process is crucial because it maintains the high osmolarity of the medullary interstitial fluid, which is essential for the kidney's ability to concentrate urine. As the concentrated filtrate ascends, it begins to dilute because electrolytes are being transported out into the medullary interstitium. This gradient is vital for the kidney's function, allowing it to produce urine that is more concentrated or dilute depending on the body's needs. So overall, these two regions work together beautifully. The descending limb concentrates the filtrate by water reabsorption, and the ascending limb dilutes it by electrolyte transport, maintaining the osmotic gradient that's key for water reabsorption elsewhere in the nephron. Let's talk about the conditional reabsorption that happens here. The tubule can reabsorb sodium ions and water depending on the body's needs, which is essential for maintaining proper fluid balance. It also has the ability to reabsorb bicarbonate ions, HCO, helping to regulate the blood's pH level. At the same time, this segment is actively secreting hydrogen ions and potassium ions, as well as ammonia. NH3, this secretion plays a crucial role in keeping the blood's pH within a healthy range and ensuring the sodium-potassium balance is maintained. Now, moving on to the distal convoluted tubule, or DCT. This part of the nephron is really important because it fine-tunes the reabsorption and secretion processes. It adjusts what gets reabsorbed or secreted based on the body's current needs, helping to regulate blood pressure, pH, and electrolyte levels. So overall, the DCT is a key player in maintaining homeostasis, making sure our blood remains balanced in terms of fluids, pH, and electrolytes, aligning perfectly with the overall theme of kidney function and fluid regulation. This long duct extends from the cortex of the kidney to the inner parts of the medulla. As it runs through this region, it plays a crucial role in the kidney's ability to concentrate urine. Large amounts of water are reabsorbed here, which helps produce the concentrated urine our bodies need to conserve water effectively. This segment also allows small amounts of urea to pass into the medullary interstitium. This process is essential because it helps maintain the osmolarity gradient, which is key for water reabsorption and urine concentration. Additionally, the collecting duct is involved in maintaining the pH and ionic balance of the blood. It does this through a selective secretion process, which helps regulate the levels of ions like hydrogen and potassium, contributing to overall homeostasis. So, in summary, the collecting duct is vital for water conservation, osmolarity regulation, and maintaining blood pH and ionic balance, highlighting its central role in the kidney's function within the broader context of fluid and electrolyte regulation in the body. Let's talk about osmoregulation. This is a critical process that organisms use to maintain the balance of water and salts in their bodies. It's essential because too much or too little water can disrupt cellular functions and overall health. Osmoregulation involves mechanisms that carefully control the movement of water and solutes across cell membranes. Different organisms have evolved various strategies to manage this, depending on their environment. For example, freshwater fish need to prevent their cells from swelling due to excess water, while marine fish work to conserve water and excrete salts. A key concept here is the role of specialized organs, like the kidneys in mammals, which filter blood and regulate water and salt levels. These organs help maintain homeostasis by adjusting urine concentration and composition. Connecting this to the broader theme of physiological regulation, osmoregulation is a prime example of how organisms adapt to their habitats to survive and thrive. It highlights the importance of internal stability, even when external conditions change dramatically. Understanding osmoregulation gives us insight into the complexity of biological systems and the incredible ways life maintains balance in a constantly changing environment. Mammals have the remarkable ability to produce concentrated urine, which is essential for conserving water. A key part of this process involves the Henle's loop and the vasa recta. These structures work together through a mechanism known as the countercurrent mechanism. In the Henle's loop, the flow of filtrate in the two limbs moves in opposite directions. That's what we call countercurrent flow. This arrangement is crucial because it allows for efficient exchange and concentration of solutes. Similarly, in the vasa recta, blood flows in a way that increases osmolarity towards the inner medullary interstitium. The blood in these vessels moves in a countercurrent fashion to the flow of filtrate, helping to maintain a high osmolarity gradient. This coordinated movement of both the filtrate in the Henle's loop and blood in the vasa recta creates a highly effective system for concentrating urine. This process, the countercurrent mechanism, is a perfect example of how anatomical specializations enable physiological efficiency, connecting back to the overall theme of how kidneys regulate water and solute balance in the body. Let's take a closer look at osmolarity. Osmolarity refers to the concentration of solute particles in a solution, specifically expressed as the number of osmoles per liter. It's a key concept because it helps us understand how fluids move across cell membranes. When osmolarity is balanced between compartments, cells maintain their shape and function properly. But if osmolarity is too high or too low, it can lead to fluid shifts that affect cell health. This is especially important in medical settings, where managing fluid and electrolyte balance is crucial. Essentially, osmolarity is a measure that ties together various solutes and helps us predict how water will behave in the body. By understanding osmolarity, we can better grasp how the body maintains homeostasis and responds to different physiological changes. NaCl, or sodium chloride, is transported primarily by the ascending limb of the loop of Henle. This segment actively moves NaCl out of the tubule into the surrounding interstitial fluid. What's fascinating is that this exchange occurs in close proximity to the descending capillaries of the vasa recta, creating a highly efficient countercurrent system. The NaCl then gets returned to the medullary interstitial fluid via the ascending capillary of the vasa recta, maintaining a crucial osmotic gradient. Similarly, small amounts of urea enter the thin segment of the ascending limb of the loop of Henle. From there, urea is also transported back into the medullary interstitial fluid through the collecting duct. This close anatomical relationship between the loop of Henle and the vasa recta, along with the countercurrent flow, is essential. It helps sustain an increasing osmolarity as you move from the cortex, which has about 300 milliosmoles per liter, towards the inner medulla, where it reaches approximately 1,200 milliosmoles per liter. The primary contributors to this osmotic gradient are NaCl and urea, and this gradient is critical for the kidney's ability to concentrate urine efficiently. This countercurrent mechanism is a perfect example of how structure and function work together to maintain homeostasis. Now, let's talk about the countercurrent mechanism. This process is crucial because it helps maintain a concentration gradient in the medullary interstitial fluid. Why is that important? Well, this gradient makes it easier for water to be absorbed from the filtrate that's present in the collecting duct. As a result, the urine becomes more concentrated. In fact, human kidneys can produce urine that is about 4 times more concentrated than the initial filtrate. The main goal here is to concentrate sodium chloride in the interstitial fluid. When sodium chloride is concentrated, it creates an osmotic pull that causes water to diffuse out of the collecting ducts. This not only concentrates the urine, but also plays a vital role in maintaining the body's fluid and electrolyte balance. So overall, the countercurrent mechanism is a finely tuned system that helps us conserve water and regulate urine concentration efficiently. As urine flows downwards in the collecting tubule, it encounters increasingly higher concentrations of solutes in the interstitium. This causes water to leave the tubule via osmosis, leading to urine concentration. Urea plays a crucial role here by moving from the interstitium into the descending limb of the loop of Henle, and then back into the interstitium through the collecting tubule, helping maintain the osmotic gradient. Meanwhile, the ascending limb of the vasa recta transports sodium chloride into the interstitium, which is essential for establishing the concentration gradient. This gradient ranges from about 300 milliosmoles in the cortex to 1,200 milliosmoles in the medulla. It's this gradient that allows the kidneys to produce concentrated urine. Sodium chloride is actively transported from the ascending limb of the loop of Henle to the vasa recta, aiding in the overall process. In summary, the formation of concentrated urine relies on this delicate interplay of solute transport and water movement, creating the necessary osmotic gradient for water reabsorption and urine concentration. Let's talk about diuresis, which is simply the increased or excessive production of urine. This process is crucial for maintaining fluid balance in the body. Now, several factors and systems work together to regulate kidney function and ensure this balance is maintained properly. One key player is the atrial natriuretic factor, or ANF, which is released by the heart. When the heart senses that blood volume is high, It releases ANF to promote the excretion of sodium and water, helping to reduce blood volume and pressure. Then we have the renin-angiotensin system, or RAS, which is activated when blood pressure drops or when blood volume decreases. Renin is released, leading to a cascade that results in vasoconstriction and water retention, both of which increase blood pressure. The juxtaglomerular apparatus, or JGA, is another critical component. It detects changes in blood pressure and sodium content within the kidney and signals adjustments accordingly. Additionally, the hormone ADH, also known as vasopressin, is secreted by the hypothalamus and released from the pituitary gland. ADH plays a vital role in controlling water reabsorption in the kidneys, making sure we retain enough water when needed or excrete more when necessary. Importantly, the hypothalamus monitors the body's hydration status and communicates with the JGA and the heart to fine-tune kidney function. So overall, the functioning of the kidney is governed by a sophisticated hormonal feedback mechanism involving the hypothalamus, the JGA, and the heart. This interconnected system ensures that fluid and electrolyte balance, blood pressure, and overall homeostasis are maintained effectively. When the body experiences excessive loss of fluid, the osmoreceptors are initially switched off. But as this loss continues, it triggers a series of responses. The osmoreceptors get stimulated, which leads to an increase in the glomerular filtration rate, or GFR. This activates the hypothalamus, and as a result, blood vessels constrict, causing an increase in blood pressure. At the same time, the pituitary gland releases antidiuretic hormone, ADH. This hormone plays a crucial role because it reduces diuresis, meaning less urine is produced, and it promotes the reabsorption of water from the tubules back into the bloodstream. Now, on the flip side, when there's an increase in body fluid, the body detects changes in blood volume, ionic concentrations, and overall body fluid volume through the osmoreceptors. The hypothalamus responds to these changes by releasing ADH, which helps restore balance. So, in essence, these mechanisms work together to maintain homeostasis by adjusting water retention and blood pressure based on the body's needs. This process highlights how tightly regulated our fluid balance is, ensuring we stay stable even with fluctuations in fluid loss or intake. Now, let's look at how the renin-angiotensin system works to regulate blood pressure. When there's a fall in glomerular blood flow or pressure, this activates the juxtaglomerular cells, also known as JGA cells. In response, they release renin, which is a key enzyme in this mechanism. Renin's role is to convert angiotensinogen, a protein produced by the liver, into angiotensin I. This is an important step because angiotensin I is relatively inactive. Next, angiotensin I is converted into angiotensin II by an enzyme called ACE, or angiotensin-converting enzyme. Angiotensin II is the main effector molecule in this system. It acts to increase the glomerular filtration rate and promotes reabsorption of sodium and water from the distal parts of the tubule in the kidney. These actions work together to increase blood volume and, consequently, blood pressure. So, essentially, when blood flow drops, this system kicks in to restore pressure by increasing blood volume and constricting blood vessels, maintaining overall blood pressure stability. This is a vital part of our body's ability to adapt to changing conditions and keep us functioning properly. When blood pressure or blood volume increases, the cardiac atria respond by releasing a special peptide called atrial natriuretic factor, or ANF for short. This hormone plays a crucial role in helping the body manage blood pressure. It causes the blood vessels to relax or vasodilate, which helps lower the blood pressure. At the same time, it keeps the renin-angiotensin system in check, preventing it from raising blood pressure again. So, in essence, ANF acts as a natural counterbalance to high blood pressure, ensuring the cardiovascular system remains in a healthy, balanced state. This mechanism highlights how the heart isn't just a pump, but also an active regulator of blood pressure through hormonal signals. Let's start by looking at how our body regulates kidney function and blood pressure. One key mechanism involves vasodilation, which is the widening of blood vessels, leading to a decrease in blood pressure. This helps reduce the workload on the heart and improves blood flow. Conversely, when needed, blood vessels constrict to increase the glomerular filtration rate, or GFR, and raise blood pressure, ensuring the kidneys get enough blood to filter effectively. Another important process is the reabsorption of water in the distal convoluted tubule and collecting duct. This step is crucial for maintaining proper fluid balance and blood pressure. When blood pressure drops, the body responds by activating systems like the renin-angiotensin system, which helps restore pressure by constricting vessels and promoting water retention. We also see the secretion of hormones like atrial natriuretic factor, or ANF, which acts to reduce blood volume and pressure by promoting salt and water excretion. On the other hand, antidiuretic hormone, or ADH, increases water reabsorption, supporting blood pressure when it drops. These processes are tightly coordinated through feedback mechanisms involving the heart, the juxtaglomerular apparatus in the kidney, and the hypothalamus. Together, they ensure our blood pressure remains within a healthy range and our kidney function is properly regulated, which is essential for overall homeostasis. Micturition is the process by which our body expels urine from the bladder. It might seem simple, but it actually involves a complex neural mechanism known as the micturition reflex. Now, urine is initially formed in the nephron, which is the functional unit of the kidney, and it gets stored in the urinary bladder until our brain signals that it's time to go. When we decide to urinate, our central nervous system sends a voluntary signal that triggers a series of events. First, the smooth muscles of the bladder contract, helping to push the urine out. At the same time, the urethral sphincter, which normally keeps urine in, relaxes. This coordinated action allows urine to be released smoothly and efficiently. Understanding this process highlights how the nervous system controls not just voluntary movements, but also involuntary reflexes that are essential for maintaining our body's balance and health. It's a perfect example of how different systems work together seamlessly within the overall theme of bodily regulation. Now, let's look at how the relaxation of the urethral sphincter plays a crucial role in the process of urination. When the bladder fills, signals are sent to the nervous system, specifically the central nervous system, or CNS. This triggers a voluntary signal that prompts the relaxation of the urethral sphincter, allowing urine to flow out. At the same time, the smooth muscles of the bladder contract, which increases the pressure inside the bladder. This coordinated action, relaxing the sphincter and contracting the bladder, ensures that urine can be released efficiently. This process is part of what's called the micturition reflex, a complex but well-orchestrated reflex involving the nephron, the basic functional unit of the kidney, which produces urine. So, in essence, the CNS integrates signals from the nephron and the bladder, and through the micturition reflex, it manages the voluntary process of releasing urine. This entire sequence highlights how our nervous system and urinary system work hand in hand to control when and how we urinate. On average, an adult human excretes about 1 to 1.5 liters of urine each day. This urine is a yellow-colored, watery fluid that is slightly acidic, with a pH around 6. It also has a characteristic odor that we can often recognize. It's interesting to note that changes in diet can cause the pH of urine to vary quite a bit, anywhere from 4.5 to 8, depending on what we eat. The yellow color we see is mainly due to a pigment called urochrome, which is a breakdown product of hemoglobin. Also, on average, about 25 to 30 grams of urea are excreted daily. These details highlight how urine reflects our body's metabolic processes and how it can vary based on our lifestyle and diet, which ties into the overall theme of understanding how our body maintains balance and health. Now, let's talk about how various metabolic disorders can influence the composition of urine. When there's a metabolic imbalance, it often shows up in the urine, making urine analysis a valuable diagnostic tool. By examining urine, clinicians can identify signs of different metabolic disorders and also assess how well the kidneys are functioning. For example, if we detect glucose in the urine, a condition known as glucosuria or ketone bodies, which is ketonuria, it can point toward diabetes mellitus. These indicators are crucial because they help us understand what's happening inside the body and guide appropriate treatment. So, in essence, urine tests are an essential part of diagnostics, providing insight into metabolic health and kidney function, and play a key role in diagnosing conditions like diabetes. Let's take a closer look at the lungs and their role in excretion. They're responsible for removing large amounts of carbon dioxide, which is about 200 milliliters per minute. That's a significant amount that our body needs to get rid of to maintain proper blood pH and gas balance. In addition to CO2, the lungs also help eliminate a considerable quantity of water every day. This process is vital for maintaining fluid balance within our body. It's important to see the lungs not just as organs for breathing, but also as key players in excretion. When we think about the overall system, other organs like the kidneys and skin also contribute to removing waste. Together, these organs work to keep our internal environment stable, which is essential for overall health and proper functioning. Recognizing the lungs' role in excretion helps us appreciate how interconnected our body systems really are. Now, let's talk about how the liver functions in secreting bile. The liver produces bile, which contains several important components. These include bilirubin and biliverdin, which are breakdown products of red blood cells, as well as cholesterol and degraded steroid hormones. The liver also processes drugs, certain vitamins, and other substances. Once produced, this bile is stored in the gallbladder and then released into the digestive tract when needed. As bile enters the alimentary canal, these substances pass along with the food, aiding in digestion and the elimination of waste products. The key point here is that the liver plays a crucial role in detoxifying the body and managing waste through bile secretion. This process highlights the liver's central role in maintaining overall health and homeostasis, especially through its involvement in digestion and waste removal. Remember, everything the liver secretes eventually makes its way out of the body along with fecal matter, completing the cycle of processing and elimination. Now, moving on to the sebaceous glands, these are responsible for producing sebum, which plays a crucial role in skin health. Sebum helps eliminate unwanted substances like sterols, hydrocarbons, and waxes from the skin surface. This process not only keeps the skin moisturized, but also provides a barrier against external pollutants and microbes. On the other hand, sweat glands produce a watery fluid that mainly contains sodium chloride, small amounts of urea, and lactic acid. This sweat isn't just about cooling the body, it's also a way to help regulate temperature and maintain homeostasis. Remember, your skin has two main types of glands, sebaceous and sweat glands, each performing specialized functions that work together to protect and regulate your body's internal environment. Understanding these glands helps us appreciate how our skin adapts and responds to daily exposure and environmental changes. Today, I want to introduce you to an important medical device called the artificial kidney. It's a machine designed to help people whose kidneys are no longer functioning properly. Essentially, its main job is to filter the blood, removing waste products like urea and other nitrogenous wastes that the kidneys would normally handle. This process is known as hemodialysis. Hemodialysis is a life-saving procedure for patients with kidney failure, helping to cleanse their blood and maintain a healthy balance of chemicals and fluids in their bodies. This device plays a crucial role in supporting the overall theme of kidney function and the ways we can assist those with impaired kidney health. Now, let's take a closer look at the process involved in dialysis. A dialyzing machine, or an artificial kidney, is connected to the patient's body. Inside this machine, there's a long cellulose tube that's surrounded by dialyzing fluid in a water bath. The process begins with drawing blood from a convenient artery, which is then pumped into the dialyzing unit. To prevent blood clotting, an anticoagulant like heparin is added. Once the blood enters the dialyzing tube, tiny pores in the tube come into play. These pores allow small molecules such as glucose, salts, and urea to pass into the water bath, effectively filtering out waste products. At the same time, larger molecules like blood cells and proteins are too big to enter these pores, so they stay in the blood. This stage is quite similar to what happens in the glomerulus during natural filtration in the kidneys. After the blood is cleaned, it's pumped back into the body through a vein. The key principle here is dialysis itself. It's essentially mimicking the kidney's filtering function to remove waste and excess substances, helping maintain the body's balance. This process is vital for patients with kidney failure, ensuring their bodies continue to function properly. Kidney transplant is considered the ultimate method for correcting acute renal failures when other treatments are not enough. It involves transferring a healthy kidney from a donor to a patient whose kidneys are no longer functioning properly. The donor kidney can come from a healthy person who has been declared brain dead, which is a common source, or from living relatives like siblings or close family members. Choosing relatives helps reduce the chances of the body rejecting the new organ because of genetic similarities. To prevent tissue rejection, patients usually receive immunosuppressive drugs, which help their immune system accept the transplanted kidney. This procedure is a significant advance in nephrology and offers hope for many patients with severe kidney problems. Let's start by looking at inflammation of the glomeruli in the kidney, which occurs when proteins or red blood cells enter the filtrate due to injury. This condition is known as glomerulonephritis or Bright's disease. It involves damage to the filtering units of the kidney, leading to impaired function. Moving on, kidney failure, also called renal failure, is a serious condition where the glomerular filtration stops entirely, meaning both kidneys stop working effectively. In such cases, a kidney transplant becomes the ultimate solution to restore function and maintain life. Next, we have renal calculi, which are essentially stones or insoluble masses made of crystallized salts that form within the kidney. These stones can cause significant pain and obstruct urine flow, making their management crucial. Finally, uremia is characterized by a high concentration of non-protein nitrogen compounds like urea, uric acid, and creatinine in the blood. When urea levels become too high, hemodialysis can be performed to remove these toxins and help stabilize the patient. All of these conditions highlight the importance of the excretory system and its critical role in maintaining our body's internal balance and overall health. Now, let's briefly touch on some specific topics related to excretion and kidney function. Hemodialysis and kidney transplants are critical treatments for those with severe kidney failure, helping to replace or support the kidney's filtering role. Moving on, micturition is simply the process of urination, which involves the coordinated activity of muscles and nerves. Disorders in this process can lead to significant health issues, so understanding their causes is important. Next, we have the functions of the tubules within the nephron. They're essential for reabsorbing nutrients, balancing ions, and concentrating urine. The human excretory system, of course, includes these kidneys along with other organs, all working together to remove waste products efficiently. The role of other organs, like the skin and lungs, complements kidney function by excreting waste in different forms, such as sweat and carbon dioxide. Regulation of kidney function is also vital. They adjust filtration rates and reabsorption based on the body's needs, maintaining homeostasis. Urine formation begins with the glomerular filtration rate, or GFR, which determines how much plasma is filtered by the kidneys. The concentration of the filtrate involves the countercurrent mechanism, a fascinating process that helps produce concentrated urine, conserving water when needed. Across the animal kingdom, excretory organs vary widely, from protonephridia in flatworms to Malpighian tubules in insects, highlighting the diversity of adaptation for waste removal. The nephron, as we've discussed, is the functional unit of the kidney, and understanding its structure helps explain how filtering, reabsorbing, and secreting occur. Finally, there's the topic of nitrogenous wastes, which differ among animals, urea, uric acid, and ammonia, depending on their habitat and evolutionary adaptations. And that wraps up the key points we've discussed today. Let's start by looking at some important hormones involved in regulation. Calcitonin is one, but it is not the one also known as antidiuretic hormone. That role belongs to vasopressin, which helps regulate water balance in the body. Adrenaline, on the other hand, is a hormone involved in the fight-or-flight response, not water regulation. Now, moving on to kidney structure. The renal corpuscle, which includes the glomerulus and Bowman's capsule, is essential for filtering blood. The glomerulus, along with Bowman's capsule, is called the renal corpuscle. The cortex of the kidney contains structures like the renal corpuscle and the proximal and distal convoluted tubules. Speaking of the nephron, the cortical region extends between the medullary pyramids and contains parts like the renal corpuscle and proximal convoluted tubule. Inside the kidney, the cortical region actually extends between the medullary pyramids as the renal pelvis, which is a crucial area for urine collection. Let's talk about some common conditions and substances. The accumulation of urea in the blood is called uremia, which indicates kidney dysfunction. Kidney stones, known as renal calculi, can also cause issues. The medullary zone of the kidney is divided into medullary pyramids that project into the calyces, important for concentrating urine. When it comes to excretion, the lungs play a key role in removing carbon dioxide, not ammonia or urea. The least toxic excretory product among those listed is urea, which is less toxic compared to ammonia or uric acid. Understanding the process of filtration, blood filtration occurs at the glomerulus in the nephron. The pH of human urine is typically around 6.5, making it slightly acidic. The primary waste gases excreted through the lungs include carbon dioxide, which is expelled during respiration. Finally, considering the excretory products in animals, ammonia is highly toxic, whereas substances like urea are less so. The body converts ammonia into urea to safely eliminate it through urine. That wraps up our review. Remember, understanding these key components helps us grasp how our body maintains internal balance and filters waste effectively.